Hello, everyone. Thank you for coming to today's webinar on best practices for non-CRF data management. Today, we'll be talking about some strategies and best practices for managing non-CRF data. My name is Erin Erginer, and I have 20 years of experience in clinical research and healthcare settings, specializing in acquisition, management, and transformation of clinical biospecimen and digital health assessment data. Over the course of my career, I have worked in various positions centered around biospecimens and the data that comes from their assessments. I've worked on both the data generation side as well as the data acquisition side. I've worked in healthcare settings, dealing with patients, collecting biospecimens, making sure samples were labeled and coded correctly. I've also worked in labs, accessioning incoming samples, performing various tests and analyses, performing QA reviews, and aggregating and transmitting results. I've worked in both CRO and large pharma settings, acquiring, managing, cleaning, programmatically transforming, and delivering non-CRF data to biostats teams for analysis and review. Over the last 20 years, I have seen a lot of things go wrong, and it's my hope that the strategies I'll cover today will help you avoid some of the mistakes I have seen in the past. These days, a ton of data is collected for each subject in a clinical trial. Gone are the days of having a few visits, a few parameters measuring safety and efficacy, trials that would be over in a matter of weeks. With the complicated trial designs that are in place today, not only are trials lasting longer with more subjects, but the types of data we're collecting at each of these trials is significantly higher than it was even 10 years ago. There are sources that are digital, including eCOA, data coming in from implantable, ingestible, and wearable devices. There's data collected for eSource, things that were done like consent, visit information, visit assessments, in addition to things that are collected as biospecimens, samples that are collected from each subject either at the site or at a satellite lab. This data includes things like biomarkers, sequencing data, all the various omics data. This data is very complicated, and the volume of data that comes in from all of these assessments is overwhelming. Programmers then take those huge data sources and extract out the relevant data points and then put them into tables and into spreadsheets. Statisticians then do some very hard math to figure out the meaning of the results that's extracted. They report them in tables, listings, and figures, and that information is then submitted to regulatory agencies for review. So why is a strategy for non-CRF data essential? The volume and complexity of data coming in from numerous external sources and vendors is drastically increasing. Over 70% of data is captured as non-CRF external data. The majority of trials have more than 4 sources of external data. For certain therapeutic areas, including oncology, that number averages closer to between 9 and 12 different sources of data. That is a lot of data transfer specifications and data transfers that have to be validated, study-specific checks that have to be created and validated. There's a lot of work put into managing this bolus of data. This means that sponsors either need to increase their headcount by getting more programmers, by getting more data standards teams and more data managers, or there needs to be a way to find a more effective workflow. Unfortunately, most of the technology in our space is currently not focused on non-EDC data. End of study timelines have increased by 32% over the last several years, and the number one cause cited for end of study delays is non-CRF data issues. Not only does this delay patients getting the treatment that they need, it can also equate to millions of dollars in additional costs for a study sponsor. In fact, the estimated average cost per day of a trial running longer than it was planned is $760,000. This is due to not only lost opportunity cost, but all of the operations that are required to maintain a study. Another reason why having a non-CRF data strategy is very important is that there are strict requirements surrounding data integrity. eCOA Plus is the acronym for the data integrity principles introduced by the FDA that are central to GXP operations in life sciences. Data has to be attributable, legible, contemporaneous, original, accurate, complete, consistent, enduring, and available. Because non-CRF data is collected from many external sources, this can be particularly challenging. You must make sure that data integrity and quality are held to the same standards as data collected in an EDC. One of the easiest ways to ensure this is to eliminate as many manual handoffs as data works through the study lifecycle to ensure proper data integrity and audit trails are in place. Data integrity can also be ensured by creating a robust risk management plan, which involves anticipating the various types of errors that might occur, assessing the probability of error frequency, and evaluating any likely consequences. For all possible data issues, you should predetermine the decisions that will be taken to mitigate those risks. Having all of these plans in place and doing the work upfront will help you ensure that the data integrity is held to the same standards that the FDA and other regulatory agencies are requiring. Data security is another component of reasons why having a strategy for managing this type of data is very important. Protecting non-CRF data from unauthorized access is crucial, especially when you are dealing with the kind of patient assessment results that are exchanged outside of the EDC. There are so many ongoing studies that collect the various omics and sequencing data, and it's important that there is a high level of caution used when transmitting such sensitive information. Included with data security are things like ensuring regulatory compliance to data security rules, ensuring that data has proper encryption, documenting the various access controls, and ensuring that there's a complete audit trail that has full traceability of each data point as it's handed off between internal and external teams and internal teams as data is changed throughout the course of the lifecycle. It's also important to remember that data isn't just a quality risk. Most of the data that is used for safety monitoring and clinical reviews stems from external vendor data. Having this data clean, correct, and available to biostats and clinical teams as soon as possible is critical to ensuring the safety of subjects. These teams aren't just looking for individual result level discrepancies and oddities. They are looking at trials and cohorts holistically. This means that the data needs to be aggregated and in a standardized format so that the trends can be evaluated. Another reason why it is so important to develop these strategies is that the future is digital. The majority of data coming in the future for clinical trials will be captured in an electronic format. There are several things that have changed in our industry in the last few years. One of them is the FDA Modernization Act 2.0, which was enacted in December of 22. This act eliminates the requirements that were set forth previously for animal testing. It opens the door to drug development using computer modeling and microphysiological systems, MPS, and the use of these alternatives to animal testing is going to increase at all stages of drug development. So as the types of trials that are using this information and using these new technologies continue to increase, we're going to see a lot more digital data coming from these types of sources. Following COVID-19, we're also seeing a lot more decentralized trials. Decentralized trials are trials that are not necessarily focused around the site location, but can be conducted using various tools, technology, and mobile laboratories to really allow patients to be participating in clinical trials from almost anywhere. Decentralized trials result in increased access to more diverse populations globally. It results in better compliance and completion rates, and therefore reduces the operational costs needed for maintaining the study. This, of course, means, though, that there are more types of data that were captured traditionally in an eCRF that are now going to be captured in various types of eSource and eConsent sources. One quick side note, if you are interested in learning more information about the FDA Modernization Act and what it means for the future of clinical trials, you can visit the Certara website. There is a great ebook available that has lots of information about what this means for our industry. Next, I'll talk through some strategies and best practices that you can implement at your organization. The first strategy we'll talk about is specialization of resources, creation of cross-departmental teams or sub-teams within data acquisition, data management, data standards, and clinical programming with a vendor data focus will really increase the speed with which you can manage this data. This team should be involved with various stages of review of the different documents used in clinical trials. The first one is the protocol. Protocol authors are traditionally concerned with the clinical needs, and secondarily, they're going to be concerned with different EDC setup needs and trial design needs. But this information is critical to be reviewed at an early stage of the protocol so that you can make sure that all of the visits and the assessments that are being collected make sense, to review for any contradictory or missing information for the various sample collections, and to ensure that there is a plan in place for any new data types that are mentioned that will be captured during the course of the study. Even in the very earliest stages of review of the draft, these teams should be involved. The earlier that these teams are involved, the faster the setup of the study will be, and it will also eliminate the typical very early post-go-live changes that occur when a study is starting up. It's especially important because the non-CRF data that's collected that's specified in the clinical protocol is often vague and incomplete. There are sometimes discrepancies and contradictions, and it's crucial that someone that understands this type of data, the sample collection and the eCOA data, reviews the protocol in the early draft stages. These teams should be responsible to make sure that there is increased clarity in the text of the protocol that really does specify what information should be collected and reduces things that are overly vague, including statements that I've seen in the past like biomarkers may contain but are not limited to. Having these teams available early on to review this information will reduce these patterns that we see over time. The non-CRF vendor acquisition team should also be involved in the vendor selection process. They should ensure that contracts are reviewed in early stages by representatives from these teams, and they should make sure that there is more detailed information about data format expectations and requirements that are added to the contracts themselves. These teams can help make sure that vendor systems are validated and that their workflows are validated and follow and comply with all GxP requirements. Make sure that there is not a reliance in the contract on something like a checkbox that says, are your systems validated, yes or no? Have a deep dive and understand exactly what that validated process is. Typically, the teams that are in procurement that are managing these contracts don't have the type of experience that's required to really understand if the processes at the vendor's location are up to the expectations of the sponsors. For all new vendors, it's very important that you make sure that an external data team representative is present in all of the early contract discussions so that you can make sure that any inefficient, unaudited workflows can be identified early on and new solutions are proposed. Data transfer agreements, or DTAs, are used between sponsors and vendor organizations. Contained within these data transfer agreements are the data transfer specifications, or DTS. These documents are created to provide the process around transferring data files, the expected formats and requirements, the data definitions and variables. All of that information must be agreed between any internal and external stakeholders before any data transfer files are completed. The review process should be conducted by every single team or department that will be impacted by any data issues or errors with the data collection set forth in the data transfer specifications. These teams may include, but are not limited to, data management, data acquisition, clinical programming, data standards, biostats and stats programming teams, as well as the clinical team who will be reviewing the different values and results. This process can take a very long time. It's typically done by communications in spreadsheets through emails. This becomes even more difficult when you're working with external teams that are going to be delivering those data files. So any of the vendor teams sending data, typically those vendor teams have reviews that are conducted by their scientists and clinical teams, by their sample management team, any project managers, as well as the team that will be doing the actual programming to deliver those files to the sponsors. All of these teams must have adequate time to review, as each has a different sort of perspective on the types of information that they're an expert in or will be responsible for providing. So it's important that all these teams are reviewing that information. The DTS should be reviewed for correctness and consistency. You should also be making sure that all of the values that are necessary for final reporting, so mapped all the way through from raw to any kind of data management review, structure and format, to SDTM, ADAM, and TLFs, all of the review should be conducted to make sure that if there are any new data points or assessments being captured, that all the information necessary to report that to regulatory agencies is contained in that source information. I've seen many times where studies start and there's missing information that's required either to do calculations or to differentiate different records. And the problem is, if you miss that information of data collection, you can't go back and get it. There isn't a way to sort of program your way out of that error. So it's very important that there's a significant amount of time put into creating data transfer specifications and having that review be done and completed by everyone. While it might delay some study startups to make sure that everyone has time to review it, it's very important to do this because any changes that are done early in these stages can have significant delays. For example, if you are working on a data transfer specification and a time point for an assessment is missed, it is possible that that same information is being used to help with the different kit creation. So each of these biospecimens that is being captured needs to have an appropriate tube and package to go into and have instructions for the site to be able to collect and transfer that information. And all this information is often tied together with that data transfer specification. If something happens where a visit is missed or a visit is added and there's a change, that impact can have significant delays on the creation of those materials. It takes typically 4 to 6 weeks to create all of the various collection kits that are required to be sent to the different sites. And so it's important, again, to make sure that any errors are caught as early on as possible to reduce the delays to any kind of study startup. Another best practice for dealing with data transfer specifications is to make sure that you have templates that can be reused either for a generic template across the entire organization, having various templates that are already created in subset that are pre-populated based on the various therapeutic areas or assessment types. For example, having one that is set up to collect RECIST data or one that is set up to collect safety labs or imaging data, or having one that's designed for oncology or the respiratory therapy areas. Any pre-work that you can do to make transfer specification libraries and templates and have those pre-populated as correctly as possible ahead of time will reduce the time it takes to do each individual study startup. It's also a good idea not just to have those data transfer agreement templates set up and those specifications predefined, but having some kind of library of each of these structures and formats in the data transfer specifications that are predefined so that you know even the various panels of tests that you might be expecting at those. That will allow you to ensure that you've got all of your test codes and your methods and categories already populated, any value level data that you've created that will help you sort of differentiate the records and provide even more detailed rules about how you expect that data to be received. Pre-building these specifications in a way that is set up as a library, something that is searchable so people can find similar studies that have used that same type of assessment or the same vendor in the past. All of these things will reduce the time it takes to get these set up. Typically, the review process should be the longest part of this data transfer specification creation and setup. So any work that you can do to make sure that the actual action and operations piece of it, the workflow to create those specifications, is very important. It's also a good idea to have a process around documenting changes to each of these data transfer specifications. This can be very challenging, again, when you are emailing this information back and forth, trying to document the different changes requested or comments added by the various teams can be tricky. So it's best to have some sort of centralized tool so that you have the ability to have collaboration and discussion so that all teams can see the questions that were asked by the various team members, and any commentary can be reviewed by everybody. It's also important to make sure that there's truly one single source of truth for a data transfer specification. These transfer specifications often change not just at the beginning of the study during the review cycle, but often during the course of the study, as the study might have any kind of protocol amendments or other changes. All of these data transfer specifications will change. There's typically multiple versions, and so having appropriate version control is extremely important when you're trying to have different things being sent to your organization. Even the smallest of changes that are happening to the specifications require re-review by different teams. It means that the vendor programming team that's going to deliver that data has to change their source code mapping information, and that, of course, would have to be validated and go through their QC process. So even the smallest of changes can have a big impact on all the teams managing this data. What can cost even more time is if that information is not correctly defined with the changes defined in the specifications, so that teams have enough time to understand the changes that are coming to them, have documented comparisons of the various programs that they've used to have the various versions programmed and mapping specifications created. So all the documentation that's required is extremely important to make sure that there is documentation of the entire audit trail from beginning to end as the data changes. The next strategy to streamline non-CRF data management is to categorize the validation, conformance, and edit checks based on when they should be managed in the workflow and by whom. The first check category is data file conformance checks, which include reviewing files received from vendors for adherence to the specification. These include data definitions like structure, format, code lists, and terms, as well as file format and basic logic checks like looking for duplicate records. Typically, the majority of these types of checks are managed in downstream legacy systems, but many of the issues found in reconciliation and content level checks are the result of data definition and mapping errors. Really, these checks should be run upstream of those systems, greatly reducing the number of errors emerging in downstream systems as a result of dirty data. Not only should these checks be run and addressed prior to delivering the files to biostats and clinical teams, they should ideally be run and immediately available to the people that can action them, the vendor programmers. Put the responsibility of running, actioning, and managing Type 1 validation checks on the vendors. If your company is following the normal workflow, vendors deliver a file to a file repository. Then someone from the data acquisition or data management team picks up the file, runs it through internal tools or study-specific SAS checks. They then review the issues and collate them into a spreadsheet, which is then emailed back to the vendor. This process takes 2 weeks on average. That means it is 2 weeks before vendors receive the feedback and can provide a corrected transfer. That also means that any teams reviewing that data in order to do transformation, reconciliation, and patient monitoring are looking at dirty data for 2 weeks. Not only is that bad for CROs and sponsors, it is also bad for vendor teams. Vendor teams have very narrow deadlines to make corrections and send updated files. Even if the errors found are minor and can be wait to be fixed in the next transfer, that reduces the time the vendor teams have to properly confirm expectations, update their programming, and validate the changes. In addition to allowing vendors to see and manage conformance checks in real time, it is best practice to use system-based checks that don't require study-specific SAS checks, programming, and validation. If issue management and communication about issues are done in a cloud-based tool, all of the documentation around issues and changes are recorded automatically by the system. This means no more emailing spreadsheets, and all internal and external teams have visibility to outstanding items. Imagine if the comments on issues were available to all biostats, data management, and clinical teams, so that there was only one place for people to confirm that issues that they see in their downstream environments were real issues or things that were going to be rectified. The second category is reconciliation checks. This is where sponsor and CRO teams confirm that all samples and assessments that were expected to have occurred per the time and events table in the protocol were actually completed and reported in the source data file. There are many schools of thought around the level to which assessments should be reconciled. There are organizations who choose to record as much detail about sample and assessment collection as possible in the EDC. This would include times, accession numbers, device IDs, etc. Some organizations capture no information in the EDC about assessment collection. These teams rely on expected assessment schedules and detailed reconciliation of the manifests received from different parts of a sample's journey. I have always recommended a more flexible, risk-based approach to reconciliation. Some of the things that you should evaluate to determine whether or not how much something should be reconciled are whether or not the sample is a primary or secondary endpoint. If it is a secondary endpoint, will this assessment be used as a primary endpoint in near future studies? Collection treatment time is the assessment specific to the time at which a subject received the treatment. Can the samples be redrawn, and are the times in these cases really important? If it is a PK sample, yes, collection time of course matters, but sometimes 5 minutes matter and sometimes it doesn't. And that is something that should be determined by the clinical team and be part of the risk mitigation plan. Another consideration is whether or not a central lab is handling sample management. Most central teams have proper sample workflow plans and oversight, and they use things like barcodes on all samples and accompanying paperwork to reduce human errors. Another consideration is whether or not a single draw is split into different assessments. And by this, I do not mean aliquots that are sent as backup samples for the same test. The more steps that someone does during collection, the more likely that there will be errors. Next is if a single sample will be sent to different labs after processing. Samples that have only part of the material used for assessments at one lab and are then shipped to a different lab have a higher tendency to have reconciliation issues simply due to the logistics and handling by multiple teams. Each assessment for each study needs to be considered individually when making sample management and reconciliation plans. In addition to weighing the risk and appropriate quality measures, reconciliation needs to be considered from a practical operations perspective. One thing that I have found that is often overlooked is the fact that there is only so much room on the label for a specimen. You cannot fit the entire visit and time point name, the subject ID, the collection date and time, the assessment type, the lab name, the aliquot number, all on a teeny tiny label. Barcodes are a great option, but the only problem is that this only works if all of the sites and all of the labs can use that same barcode system. So this is really only practical if you are working with central labs and large site organizations. Where this doesn't work, I have always recommended the approach of using a 3 or 4 digit sample ID that serves as a code decode for a lot of that information. To implement this, you would create a spreadsheet that lists out all of the assessments and all of the time points, one tab for each assessment category. Then create subsections within those assessment types as necessary based on the number and complexity of assessments in the study. For example, you could have one tab of the sample ID spreadsheet for PK data and sections within that tab for blood samples and urine samples. Or you could have sections for PK metabolite A, blood, PK metabolite A, urine, PK metabolite B, blood, etc. Each of these different sections should have different predictable sets of numbers assigned to them. So PK metabolite A blood pre-dose would be numbered 100, 15 minutes would be 101, and 30 minutes would be 102. PK metabolite A urine pre-dose starts at 201, and so forth. The exact grouping and numbering will be completely study specific, but now you can have a simple 3 or 4 digit number that can be easily fit onto a tube, easily added to accession forms, EDC extraction programming so that it can be included in datasets that are used for reconciliation. And it's easy for the site staff to quickly assess kits and collection containers so that errors are reduced. Also, it's easy to identify discrepancies when you are reviewing result files or sample manifests. Lastly, there are checks that should be conducted by the clinical team. Clinical teams typically use various software tools to assess trends across treatment groups and cohorts, as well as look for overall trends in individual subjects across the course of a trial. This evaluation can only be done with any kind of efficiency and quality assurance after data is aggregated and in a standardized format. These in-stream reviews of safety and efficacy are critical to the success of a study and to the health of the subjects participating in that study. These decisions need to be made quickly, and the teams making the decisions need to be able to trust the data that they are looking at. It is imperative the teams are only reviewing data that has been subjected to the first two types of checks. The faster data files move through the first two check categories, the faster clinical teams can do their review. And the only way to get data files through the first two categories quickly is to place a significant amount of effort on study startup tasks related to data acquisition and sample management. The next strategy we'll cover are KPIs, measuring what matters. It takes so much effort to identify, validate, select, and set up contracts with a third-party vendor. Even once a relationship is established with a lab, you need to make sure that they're complying with the terms in the contract. In order to do that, you need objective data. KPIs are necessary for making objective assessments of vendor selection, process improvements, and resourcing. Automation of valuable metrics reporting is only possible if the process is managed in a cloud-based platform. The information captured by the system serves to generate metrics for reporting that really does enable objective decision-making. Data collection standards are a set of guidelines that are established to ensure consistency and accuracy in the collection, management, and analysis of data in clinical trials. These standards are essential because they help to ensure that the data collected is of high quality, reliable, and consistent across different sites, vendors, and studies. Here are some reasons why data collection standards are necessary in clinical trials. External vendor data, including laboratory and eCOA data, may be collected using different methods, tools, and platforms. Non-CRF data needs to be aggregated with the rest of the study data into the study database to ensure that it is complete and accurate. Data collection standards can help to facilitate this integration by providing guidelines on how to format and structure the data so that it can be easily integrated into the study database. Data collection standards can help to ensure the quality of external vendor data by providing guidelines on how to collect, manage, and analyze the data. This can help minimize errors and inconsistencies, reducing the risk of inaccurate or incomplete data. By using standardized methods and procedures, data collection standards can help to reduce the time and resources required to collect and manage external vendor data. This can lead to more efficient data management workflows, reduced costs, and faster data integration and analysis. In addition to the benefits of data collection standards for CROs and sponsors, lab and digital health technology vendors also want standards. Imagine being a vendor performing the same assessments with the same methods and same parameters, but having to report that information to each data acquisition team in different ways. The cost of having programming teams, oversight, validation, and QC workflows, as well as the software and IT teams to support that technology, continues to increase for vendors. Having standards that were used by most CROs and sponsors would greatly reduce their operational costs and, most importantly, increase the speed with which they could deliver data files. Despite the clear benefits of using standards for data collection, besides the limited C- guidance, there are no standards for non-CRF data assessments. Despite a few attempts in the past to align on the creation of these, there is no industry group leading their development. We only have standards related to the format in which it needs to be delivered for submission. An example that's relatable as the fall industry conferences are approaching, it's like everyone is staying at the same hotel and needs to get to the same conference center, but people are traveling by completely different routes and completely different modes of transportation. Even in the absence of industry standards, it is extremely important for organizations to develop and maintain their own non-CRF data collection standards. Establishing internal standards for various vendors and assessment types increases reusability of data transfer specifications, reducing the time it takes to create study specs. Data collection standards will also reduce the number of study-specific edit checks that need to be programmed and validated. All of the work can be done one time to ensure transformations and mappings are set up in a way that can be automated, with new programming only required for new data types. Standards for non-CRF data should be created for all assessment types and for all vendors that send data. For central labs, these can be established in master service agreements. For smaller labs, each time a new vendor is onboarded or an established vendor is doing a new assessment, the specialized teams should work with all stakeholders to fully understand the content. Once there is review by the data management, data acquisition, data standards, clinical programming, biostats, and clinical teams on that structure and format that the data will be received in, this means that any associated listings, clinical review reports, and transformation code can be reused. The best case scenario for non-CRF data is that the vendor with whom you are working can deliver to an SDTM light data format. The closer data is to SDTM when it is received, the easier it is to map and drive automation. This is a completely reasonable expectation when you are receiving data from a large central lab with lots of programming and technology support. For niche academic labs, this is not reasonable. These small teams of scientists don't have programmers. If you are asking these teams to deliver data in SDTM-like formats, you can be sure that they are just copying and pasting data from their source LIMS systems. Going back to earlier recommendations about data integrity, this is less than ideal. For these teams, it is much better to get the data as it comes in from their systems. Being able to manage the transformation programming on the CRO and sponsor side will have way better data integrity. It will also mean that data files will come much faster from these smaller labs and will be of much higher quality. But even if the data you are receiving from them is not SDTM-like, you should still establish data collection standards for that data. It takes a significant amount of work to understand and interpret new assessments and to make sure that everything needed for mapping and clinical reviews is in place. Do that work one time really well and turn that into a standard that can be used by future teams on future studies. Another challenge with data standards is underdeveloped technology for creating and maintaining metadata. There are some generic solutions on the market that a few organizations have modified to use for clinical trials data, but often organizations just use Excel spreadsheets. This, of course, is not ideal as storing and maintaining data in spreadsheets is plagued by human error. They don't have appropriate change management and governance controls and don't make collaboration and communication of changes easy. If this is something that your company is struggling with, don't worry, Pinnacle 21 has been developing a metadata management tool for data standards that includes non-CRF data standards management capabilities. Our first version will be available in our 6.0 winter release this year. If you have any questions about this or are interested in becoming an early adopter, please reach out to me after this webinar and I can provide some more information. Currently available in production is Pinnacle 21 Data Exchange. It is a solution that is designed to streamline external data management. Data Exchange reduces timelines for vendor delivery, the timelines between collection and analysis, and the time between last subject, last visit, and database freeze. It reduces the number of resources required to deal with this type of data, and therefore it does reduce the cost of operations. At the same time, Data Exchange improves the quality of deliverables that are coming from vendors. It allows you to have more flexibility when specifying data requirements. It improves the workflows, not only for sponsors and CROs, but also for vendors. And it improves the ability to do objective decision-making using system-created metrics. With Data Exchange, you can accelerate the flow of clinical trial data coming from labs and digital sources so that better decisions are made faster. You can harness automation and reusability for processing source data. It allows you to automate oversight with executive dashboards and system analytics. You can ensure integrity with cloud-based validation and structural checks. And there's cloud-based teamwork and centralized communication software available as well. The Certara Pinnacle 21 Data Sciences platform is an end-to-end solution that improves data quality and streamlines the data management process throughout the study lifecycle. It ensures that your submission timelines aren't derailed by poor quality data, which causes costly delays, or worse, delaying getting your treatments to patients that are in need. Foundational Pinnacle 21 Enterprise helps you transform data and helps with your submission to regulatory agencies. However, in order to get to that point, you need to ensure that the data coming in upstream of that solution is in standardized format. The earlier data is standardized in the lifecycle, the more you understand the types of data you're going to be receiving, and it's going to be much easier to support the transformation to regulatory requirements. Thank you so much for listening today. Let's take a look at the questions in the chat. Vendors are used to getting issues in our issues spreadsheet. Won't they push back if they do issue management in our system? Good question. Process changes are hard. Change is always hard when you're working with something where you've got an established workflow with a vendor and you've got established templates and issue spreadsheets that they're used to using that get emailed back and forth. And there might be a little bit of hesitancy because it's a change to their system and the way that they've been working together. But what I found so far is that vendors are very open to some changes in this space. The amount of time that it takes them to navigate issues in a spreadsheet and having to wait those couple of weeks in order to get those issues is significant. And they do get a lot of pushback in terms of how long it takes them to correct those issues. And that can cause a lot of issues between the relationship between the vendor and the sponsor. And this way of working is something that, again, is a difference to what they've been used to. But once you explain sort of the new process and demonstrate how much easier it will be to have spreadsheet issues in a spreadsheet done instead in a cloud where there's one single source of truth, everyone can see the issues, it's going to be a lot easier for them to navigate. So they can have oversight themselves on how well, the project managers on that side can have oversight of how well their teams are doing, how quickly they're responding to things, as well as look at things where if you've got an issue that was raised previously and then it was closed and then it's reopened again, these are kind of things that people on both sides of the table want to make sure that are getting addressed correctly. And having them done in a system is a lot easier than trying to navigate tons and tons of spreadsheets and versioning and things like that. It will actually take them less time to do this work because if the system is set up correctly, you can do things to highlight issues that are more important than other issues. Any responses that they have, they're not going to be receiving email feedback, loops of feedback between the different teams. You can have all the communication centralized in one area so that even if somebody on one of the teams on either side was not involved in that earlier communication, everyone can go into this one single system and find out the current status of the issue, what's being done to address it. And that's regardless if the issue is due to a vendor mapping error or an error in the specification. Everyone can see what those issues are and then figure out the best way to address them collaboratively. And I think that's something where it really will help build up the relationship between the vendors and sponsors and make it so that everyone's sort of working towards that same goal. The other thing is also just having the audit trail in place so that it's easy to see the entire history of changes so that that's available to everyone and no one's having to sort of export out all the versions of a spreadsheet and export out all the different versions of specifications, things like that. If all that information is captured by the system, then any documentation requirements on either side are sort of mitigated by the system doing all that hard work for you. So I think that's something where it does provide a lot of benefit to the vendor. So even if it's a change, I think it's something that will make it easier for them in the long run, and it will definitely save them time. And again, that's easy to communicate to the people with whom you're negotiating contracts. They're going to care about the cost of the resources. And I think it's pretty easy to demonstrate how their costs can be reduced by using this new process as well. Okay, next question. How do you get all of the data for the KPIs that you mentioned? Okay, That is very difficult to do when you are doing all of the various different pieces of the process in different applications. So if you have one tool that is doing your specification creation, you're using emails to document the compliance and the agreement with and the finalization of the specifications, and then a separate tool for vendor data delivery. So they're uploading to either a file share or some sort of SFTP delivery. Again, especially if you're using multiple ways of those data files coming in, and then a separate tool in order to do all the different pieces of conformance checks if you've got a tool or just SAS checks to do the validation checks. All of those things that I mentioned, it's likely that they're not being done within the same system, and it's possible that they don't all communicate well together. So that's very, very difficult. So the best practice then would be to have one system where you can create requirements and also complete that validation and do issue management. That way you have all the pieces of information that you need in one location. Some of the key pieces of information that you'd need to have the best KPIs would be, the first one is dates. So when was the data transfer specification created? When was it finalized? When did you receive that first test transfer? And then when was the last, how many test transfers did it take? And then dates around comparing your final assessment data coming in and then you receiving that final transfer for the study. All those dates are things that hopefully you can pull out of your system in order to really have the metrics that you need. In addition to dates, you also need information about the quality of the file. And this one is a little bit tricky because sometimes without good tools, it can be a little bit subjective, and you really need to have an objective measure of quality. In our data exchange tool, we do have a progress score we call it a progress score. It looks at how well a file complies with the requirements that are built in the spec. And it's something that you can use to compare then across vendors. So if you've got a vendor that's maybe really good at assessment A, but another vendor is better at assessment B, you can sort of collate that data, pull it all together, put it in some dashboards, and figure out with the procurement team and the rest of the operations team and try to determine, is maybe the difference between having, one vendor, do you want to have multiple vendors, or is it just a better cost savings to have one vendor who is maybe not as great as at all the assessments doing the work? Is it just easier to have a better, larger contract with a single vendor? In addition, it's important to take a look at the quality and timelines that are not just within each vendor and comparing across vendors, but it's also useful to sort of do an internal look and see how there are differences across your organization. Maybe one therapeutic area or maybe early phase versus late phase are getting better results than another one, and then you can take a look at that data and figure out, do a little deeper dive and see what's the cause of that. Are they using different processes? Does one team maybe use a specialized vendor team and another one has the data transfer specs created by a regular steady data management lead? It's a really good opportunity to sort of do an internal review and determine where maybe some improvements can be made within your own workflows and processes and align better across therapeutic areas and phases or whatever the grouping of the departments might be to speed up those timelines across the board. Other pieces of information that are helpful for gathering all the KPIs that are needed is, one is the contract sign date. So I mentioned earlier that the best time to start your data transfer specification is when your protocols are finalized or the contracts are finalized with the vendor. But those vendor contract dates are typically found in even another system, typically with procurement. So if you have a way of pulling in the pieces of information from that procurement department, it's a really good date to have. So you know how long from the contract sign date is it until you've got good specs in their hand, final specs in their hand, and how soon after that are you able to get some result data coming in, even if they're test transfers. The other one would be that I do recommend that you take all the information from these KPIs and put it into a dashboard or a report, because typically the information that you have in all the different KPIs, you need to report those to leadership teams or other stakeholders. And usually when they ask for it is when they want it. And if all that information is in a lot of different systems, it takes a lot of time to pull that together. So the best idea is to have all that information in one place so you can have dashboards and reports that you can easily export and deliver as soon as that information is requested. So good question. Next question. There is a CDISC LB data collection format. Why can't we use that across the industry? I am familiar with the CDISC LB domain, and one of the advantages of this domain is that it is SDTM aligned. So that means that transformation between this format and the next step, which is typically SDTM, can be done very, very quickly. But this is I think it's about 12 years old, maybe at this point, that domain and structure. It's massive. There's something like over 100 variables. And the majority of those variables are not needed and are not applicable to each of the different studies. It's sort of a giant superset of everything you might need for a study. One of the things that takes the longest in study startup is trying to determine what things are actually required for each of the study specific needs. So I do think that it's easier to have something where you've got standards or pseudo standards that are very closely aligned to having more of them that are more closely aligned to the exact assessment types that you're getting, so that you have already sort of pre-subset all of that information into something that can be used very, very quickly for study startup. Also, the LB domain was the lab collection domain was not created democratically. So I don't think that it didn't take into account enough of the vendors' perspectives and the sponsors' perspectives when it was created. So it was done, I think, with the idea that it would be created and then there'd be feedback and then there'd be different iterations. But unfortunately, there haven't been enough resources put into that project. So it's not the most useful for most applications. It's pretty okay if you're using it for a large central lab, but it's definitely not something that I would recommend for mid and small size vendors. It's just too overwhelming. If you send that spreadsheet to them with hundreds of rows or hundreds of columns in it, the amount of time and back and forth is just not going to be worth it. So I think there are better ways of having some data collection standardization formats, and but it's a good start, and hopefully there can be some movement within the industry, various nonprofit organizations that can start to have a more democratically created set of standards and pseudo standards that are used for non-CRF data collection. Next question. How can we move away from detailed data definitions and specs and bring data in from the source and use AI to automate the mapping? I love AI questions. AI, specifically machine learning, can be used to drive mapping and automation and mapping, but only if it is data that you've received in similar structures and formats in the past. And that's just not really realistic. You're not really saving much time if you're just mapping data that you receive consistently and regularly. Even if if your organization uses central labs, it's likely that you are using more than one central lab, and it does not help with all of that, the novel data sources and assessment types that you're getting, especially from those mid and small tier labs. Just taking a look at it from the perspective of controlled terminology, so much of the data that is received for this non-CRF data does not have CDISC terms available yet. So typically sponsors will create their own terms and then update them later if and when CDISC terms become available. And often what happens is that the sponsors take the terminology they create and then build up value level metadata and biomedical concepts groupings around these terms. And all of that has to change once there are new terms and new requirements, whatever is dictated by the appropriate regulatory agencies. So AI ML is great if it's seen something similar and you want to map it exactly as you did before. So it works really, really well for things that are captured in the EDC traditionally. So vital signs, demography, things that it's done over and over and over again. It's well established. But non-CRF data is so complex and it grows faster than anyone really knows how to handle it. And figuring out what data means and where to map it will, at least with the technology that's available now, require, as far as I'm concerned, it'll always require a large effort by the experts that understand that data and really know what it means. So I think it's much faster to build automation when you've got detailed data specs and requirements and you implement tools and processes that facilitate the reuse of those specifications. I think that's the fastest way to drive automation. Again, as with the technology that's available now, I don't think that it's realistic to think that anywhere in the next 5 to 10 years we'll have something that can just take in a data file from the source data from a vendor and just automatically map it, especially when it's going to be likely containing lots of brand new information. So maybe in the future, hopefully I'm wrong. Maybe technology will grow faster than I'm anticipating, but I think that the best practice really is to use standards-driven automation in order to get the data to go through that life cycle much quicker. All right. Next question is real-world evidence data, RWE data, a type of non-CRF data? How should we manage that? So our real-world evidence data is definitely a hot topic. As of now, there is very little official guidance from regulatory agencies. And I won't speak on behalf of any regulatory agencies. Obviously, I don't work for a regulatory agency. But I did hear some good presentations at CDISC EU earlier, I think at the end of last year, maybe earlier this year. And there was an FDA representative there that made the statement that if you are submitting RWE data, it should be following the same sort of process and requirements that you would for any other data that you're submitting for a trial. Obviously, that's tricky because there are certain things that are just inherently different about RWE data versus data that's captured in a normal clinical trial. Essentially, we would need some modified data domains in order to account for those differences. I know that there is progress being made with the different agencies and organizations trying to make data domains that have those differences displayed and have, I think, maybe some new domains that they're looking at. There's also a great project that is ongoing at the EMEA, where they are looking at data from different EHR systems and seeing if they can use the current CDISC models for managing data from EHR systems, or if HL7 is really the best way to go for that. And if they did decide to use HL7, then can the tools that the agencies use currently to do data reviews and data fitness, can they use those same tools, or do there need to be modifications for them to use HL7 in combination or in replacement of some of the CDISC domains? But again, this highlights really the need to have some of these best practices in place for dealing with non-CRF data, because RWE data is a type of non-CRF data. And with all the new technologies and the changing legislation to allow us to use more information from EHRs and computer modeling in submissions means that RWE data is just going to contribute to the volume of data captured outside of the EDC to be significantly higher than what's captured in the EDC. So I think as we're waiting for some of these regulatory changes around this type of data, now is the best time to really start building up the workflow, processes, tools, technology, whatever you need to do internally in order to stay ahead of the proliferation of this kind of data. Unfortunately, that is all the time that we have for today. Thank you all so much for coming. And if you have any questions, please reach out to me. If you are interested in hearing more about our solutions for data management or data standards, please reach out to me. I look forward to hearing from you.